Hello everyone. I am further analyzing the RNA-seq data to compare gene expression in breast cancer triple-negative cells MDA231 with gene expression in estrogen receptor-positive cells MCF7. In the part 1 of this analysis, I uploaded FASTQ files from Gene Expression Omnibus GSE 188914. 3 FASTQ files from MDA231 and 3 from MCF7 were uploaded and submitted to the pre-existing workflow within the geneXplain platform. This pipeline includes subread align, feature counts, and Lima-Wurm method to process the FASTQ files. Today I will show the workflow output files. In my project, there is this whole folder with the results produced automatically by the workflow. Every sample is represented by a separate subfolder. Let's have a closer look to some of the resulting tables. Let's open the alignments log. Here the results of the method subread are shown. As shown here, the total number of reads are over 43 billion and the reads that are mapped are over 39 billion. This is about 91% of the reads are aligned and mapped to the reference genome. This is a very good quality of alignment. Alignment of FASTQ files also produces BAM files. These files can be visualized in the Genome browser as shown here. Within the Genome browser, we can toggle and view the genes from different chromosomes. We can specify the position number and scroll forward or backward. We can also copy-paste a particular ensemble ID and directly view the results for that particular genomic region. In the tree, BAM files are shown with a distinct icon, like 2 arrows. This icon is used here in the platform for all tracks. I have now added one more track with known genome variations from database SNP for visual comparisons. The ensemble genes can be visualized in the gene track. Here gene ETS1 is shown, which is located on the negative strand. Pink parts are the exons and the black parts are the introns. I zoom in to see more details, even to the level of particular nucleotides. BAM files are mapped to the entire gene, including exons and introns. Here we can also observe that some reads are variations. For example, here it is a C instead of A, which coincides with this variation RS2230004. And here there is a G instead of A, which coincides with this variation RS113082559. Interestingly, these variations are located in the 5' region of the gene ETS1. Now let's open FASTQ counts file. This table contains close to 58,000 ensemble IDs. We can see how many BAM files are mapped to each gene, including exons and introns. When we sort by number of counts, we can see many genes with 0 BAM files mapped. But when we sort with another direction, we can see many genes with quite high number of counts mapped. Such resulting files are available for each of the input FASTQ files. To summarize the alignment and feature counts, we open the FASTQ counts log. As shown here, the number of successfully assigned alignments is close to 35 billion, which is 81% of all mapped reads. To summarize the statistics, over 35 billion out of all reads are mapped to the reference genome and assigned to a particular ensemble gene. And after the gene counts, the LogFold change and p-values are calculated using the Lima-Wurm method. As shown in the graph here, the mean variance trend shows how many genes are located near the mean variance graph, which shows the number of differentially expressed genes is quite significant. Here is the table of genes with calculated LogFold change and p-values annotated with gene symbol and gene description. There are close to 26,000 genes. We can sort and filter this table by log-fold change, p-value or adjacent p-value. I now have a list of upregulated genes, downregulated genes, and non-changed genes. In this data, there is a significant number of upregulated genes. Sorting by log-fold change shows top highly upregulated genes, for example, SRGN, ETS1, CNN3, CDCP1. Sorting by p-value and adjacent p-value show that the calculations are highly significant. I will work further to map upregulated genes to geoterms, to diseases, to pathways, as well as to select a functional group of genes for the upstream analysis. This is coming in the next parts.